Well, welcome everyone. Thank you for joining us this evening for this next edition of BGSU's Science Cafe. My name is Timothy Davis, and I'm the director for the Center for Great Lakes and Watershed Studies here at the university. And for those of you who are first-time participants in Science Cafe, I host this event with my colleague Kevin McClooney from the Department of Biological Sciences as well. And the idea behind a Science Cafe is that we, in the center and the department, wanted to relay and communicate the science that we're conducting on the Great Lakes and the watersheds that surround them to you, the public that either live in Bowling Green or Northwest Ohio or wherever you're located tuning in. We just feel that it's really important that we not only share our science with our colleagues, but also with the people whose our science can impact, whether it's through fishing or recreation or otherwise. So the way that science cafes are meant to be is that we would ideally like to be in person, where we could meet as a group and have kind of an informal discussion. So the way that the current setup is, it's a bit more formal than what we had originally intended, but with everything going on in terms of in-person gatherings, obviously this is the current format. So Dr. Jeff Miner is our presenter today, and I'll introduce him in a few minutes after a few more announcements. But the way that this event works is that Dr. Miner will give us about a 20 to 25 minute presentation on his research. And then we move into question and answer time. And we like this really to be a discussion. Now, you can either, we'd ask that you hold all your questions to that time so we can have a robust discussion then. Or if you don't want to ask a question but still have a question, you can send that question either to the group at large or directly to Kevin McClooney using the chat function. So those are two ways to ask questions, but we really want a robust discussion. And so Science Cafe is really a production that takes a team of people. It's a collaboration between the Center for Great Lakes and Watershed Studies and the Center for Public Impact. So I always like to thank the director, V. Rosser, and S.D. Miller, who is really the point person for all of this. She makes a lot of the communication and the logistics happen. So we like to thank her, even though she's not able to join us tonight. But also we have a couple of students, Maisie Davis and Alessia Saul. We thank Tom Cummings from WBGU for all the technical assistance, and Pete Fairburn for all the media and graphics. We ask that you stay muted for the entire talk. And before we get going, I don't like to take up too much time because I like to really highlight the speaker, but we are beginning another, we're adding an addition to a Science Cafe. So we have a PhD student in the Department of Biological Sciences. Her name is Carrie Ritzenthaler. And she is conducting research on how learning about the environment through events such as Science Cafes impact those who attend. So we're going to announce this at the end, and in a follow-up email, ST is going to send you a link, and there's also going to be a link in the last slide in Dr. Miner's presentation. But it goes to a survey that we would really appreciate everyone filling out. It takes about 10 minutes to complete, and if you complete the survey, you'll be entered into a drawing to win one of 4 Amazon gift cards. So there is a little bit of a incentive there. But we really, Carrie's dissertation research relies on these types of responses. So we would really appreciate if you could fill out that questionnaire. Everything is anonymous. We do not collect any personal information. It's only the responses. So with that, I'm really pleased to introduce Dr. Miner. Dr. Miner is a professor in the Department of Biological Sciences, and he leads the Aquatic Ecology and Fisheries Lab. We have a great panel for you tonight who's going to join Dr. Miner after his presentation, and my colleague Kevin McClooney is going to introduce the panelists at the end of the talk. But Dr. Miner's research is really focused on invasive species and introductions into Lake Erie. And also, he really highlights, his research really centers around environmental conditions in wetlands and bays that impact important fisheries like northern pike and walleye, which are obviously a very important sport fish in Lake Erie. So we're really pleased to have Dr. Minus join us tonight. I think the presentation is going to be fantastic, and I'm really looking forward to the discussion afterwards. So with that, I'll hand it over to Dr. Miner, and I will stop talking. Thank you again for joining us.
Okay, I guess I'm ready. Hello, everybody. I'm really happy to be here today. I'm going to give you a summary of the research that primarily 2 PhD students have been conducting in my lab over the past 4 years. We do have, as Tim said, a fair amount of past research looking at invasive species, from zebra mussels to gobies to grass carp. But let's focus on these 2 species here. The walleye, which you all know because you've watched people fishing for them as you cross the Maumee River, or northern pike, which are, I'll say, ecologically very much like saltwater barracudas because they eat primarily fish and have that body shape. So let me move quickly through this so I get to what I think is some really interesting work that these 2 PhD students have uncovered. So there we go. Just a little introduction. A lot of money has been placed into the Great Lakes Restoration Initiative from the federal government to promote the restoration of habitats, environments associated with the Great Lakes. And the Ohio Department of Higher Education has also chipped in a large amount of money to try to look at how nutrients and runoff from watersheds has affected the water quality, primarily for humans, but also for the rest of the ecological community. So what do we want to do to try to get this transition from this muddy stream that we see up in the upper left-hand corner to something that is more aesthetically pleasing, more ecologically, let's call it natural? We need to reduce nutrient loading to Lake Erie. That's part of this whole restoration process. We need to reduce sedimentation. So the sediments carry a lot of the nutrients, but they carry them out into Lake Erie. Dr. Mackey, who will be on later, might be able to explain a little bit about the laws that are going into effect to prevent or to limit the amount of sediments that get pushed out into Lake Erie, primarily from dredging in our harbors. And then really what we want to do also as ecologists is to try to improve what's called ecological services, which basically means the functioning of the community or the ecosystem. We want it from a human perspective because we know that pristine or improved wetlands will sequester more nutrients and therefore keep those from going out into Lake Erie. They also provide a lot of heterogeneity in the environment, as you can see in the picture to the right, so that you almost always get higher species diversity in systems like that. And for me, it's the environmental variables that fluctuate that cause the shifts in communities of fishes or in populations and how well they do that really is interesting. And I'd like to tell you a little story about the 2 species that I listed before. First, let me say that Lake Erie is really interesting because it's shallow. Therefore, it warms up quicker than the other Great Lakes. Also, light penetrates down further, so you have more photosynthesis from algae, so you have more primary productivity, food resources for other organisms to use, and we have the Maumee and the Sandusky watersheds here, which are part of the large agricultural landscape that we have around here. And just pulling out a few facts, the Great Lakes tributaries deposit nutrients into the lakes, but about 50% of all the nitrogen that gets deposited into the Great Lakes comes from tributaries of Lake Erie. And just recently in a publication by a fellow by the name of Jay Martin from Ohio State, he showed that in fact over 50% of all of the phosphorus that gets pumped into Lake Erie through the Maumee River occurs from inorganic fertilizers. So that means that the fertilizers that farmers or you put on your lawns or your fields, not from the manure. He was trying to separate that out, what comes from manure in terms of cows and pigs and chickens that get raised here. And so it's trying to identify what the sources of the nutrients are that get into our system that make it so productive, both positively and negatively, is what we're all trying to sort of explore. Okay, so that's my nod to nutrients. I want to move on to fish. So let's talk about walleye today. For people who have been fishing for walleye, you know that you can go fishing on the Maumee River, which is here from Toledo upstream to where these fish go to spawn in the spring. They're there now. Other places that they spawn are out in reefs in the southern shores here of Lake Erie and up in the Sandusky River system, highlighted here by my laser pointer. So there are three different primary sources of these walleye, and I've studied the ones on the Maumee River. We've started focusing on the Sandusky River, primarily because of the fact that we know that these fish, when they spawn upstream, the young move downstream and move into this, I'll call it a bathtub, a fabulous nursery ground for young fish to use. So my interests are in how the biotic or the organisms in a system and the environmental variability that occurs in systems like Sandusky Bay can have implications for the growth of these small fish that are somewhere in the neighborhood of 3/8 of an inch to 1/2 an inch to 3/4 of an inch out in the lake. So we're used to thinking of walleye as fish that we try to catch, large individuals, and we all line up on the shores and try to catch them as they're out there trying to reproduce, okay? But I'm focusing on the larvae today. It's these guys. The ones on the right, the smallest one here has just hatched. It's got its yolk sac here, and it is feeding on its yolk right now. There'll be a time when after several days, when it uses that yolk up, it'll look more like this above fish here, and that's the time when it has to start feeding, okay? If it survives, and very, very few of them survive, to this size over here, which is about 1 inch to 1.5 inches, that's when they're going to start feeding on things like fish and other bigger prey that they can allow them to grow very fast in the Maumee River. So what we know about fish, generally fish, is that they are very fecund. A single female walleye will have tens or even 100,000 or more eggs. She will release those. Those will then go into the interstices or among the gravel in the area where she has laid them. The male will fertilize those. They will then incubate for a period of time, and when they hatch out, they have to use that yolk, but then they have to feed. And this time of switching to feeding is called a critical period because they have to make that transition. They are very they don't have a lot of mass. They need to get food right away, and so they only have a short period of time to be able to find some food to eat it. And so we've been looking at, along with others, the prey availability for these larval fishes, as well as people have been looking at how environmental conditions sort of force them into different locations, or it may cool down, slow down their metabolic rate, and all these factors lead to the very high mortality that we see in these organisms. So if you need a number, you might say that we start off with 1 million, and by the time they get to a size that maybe is 1 inch long, maybe there'll be 1,000 of them left. So we've just lost 99% of them. That's just a number. It can vary some, but just to think about the high rate of mortality that occurs in these systems. And so I'm very interested in trying to look at how the variability in the environment and the variability in the food resources affects the survivorship of these and growth rates of these organisms. So, as I said, they spawn in rivers. They stay in the eggs, embryos, for about what's called 70 degree days. Degree days, because they are called ectotherms, they're cold-blooded. Their metabolic rates are totally dependent upon the temperature and the environment. And so if it's warm, they will develop quickly. If it's cold, they'll develop slowly. But once they finally get to the point where they hatch, they'll stay in the rocks for a while, and then they'll have used up their yolk, and it's time to leave. And so they will now start drifting down a river, or if they're out on a reef, drifting with the current across Lake Erie. So if we think about Sandusky Bay, and for those of you who don't remember, Sandusky Bay is right about here, kind of halfway between Toledo and Cleveland. Sandusky Bay is this system where you have a river where the walleye hatch out of eggs. They drift down the river about 25 kilometers or 18 miles, and then they come out into the bay, which is, I'm going to call this the upper bay here, and then in time, as if they survive and grow, they will slowly move out into the lower bay and then ultimately leave and go out into Lake Erie and continue their life out there. So we want to look at the dynamics that occur when they're in this stage here, as they move out and try to move into the bay to try to find food, to try to find, to be able to grow fast and survive. Because if they get to that nursery, they're going to have now gone through an important bottleneck to be able to survive. So what we're going to do here is I'm going to talk about how they disperse in this system. I'm going to talk about the patterns of distribution of their prey, which we call zooplankton, and I'll show you a picture of those. And then once I get through that and show you, this is Chris Kemp's dissertation work. He has some really cool results, I think. He's going to, where we also are getting into some modeling, where we're trying to look at, can we model the dispersion of these larvae under different hydrologic conditions. So Chris organized this so that we went to this part of Sandusky Bay. He wanted to set up a sampling regime, so he took 4 different sectors here and decided he'd sample from the north to the middle to the south side to collect these larval fish. We use a net here to collect them. We collect zooplankton at the same time, their prey, so that we can relate the abundance and distribution of the larval fish with their zooplankton prey. So keep this in mind as we go here, these different colors, okay, because it'll help when we're looking at the figures. So we want to look at their prey first. May 16th, 2018 was the first day that we saw larval fish in the lake. So they were coming down this way, and we saw them in these yellow boxed areas here, okay? But what does their prey look like at that time when they're just getting, these little fish are getting to the river and it's time to eat? OMG, the density of their prey is incredibly low. Matter of fact, it's as much as 5 or 6 times lower than the area over here in this green region. So you might ask, well, why don't they just swim over there? But these guys are plankton. They just float in the water. And so they can't just decide to get up and swim a kilometer or 2 to get over there. They have to be transported slowly with a mass of water to that region. Okay? So what we see here is that it's a tough time for any little larval fish if they're stayed in this habitat. And in fact, that's what Chris found, is that the larvae were at very high densities. They were blown, we think, into this lower region of this part of Sandusky Bay where the density of prey was incredibly low. Matter of fact, Chris did some experiments and he was using that density to see how fast they would grow, and they don't grow well at all. Okay? So now we see this pattern that occurs here where the larvae are down here, but there's not food. That's really, really important. Okay? As a matter of fact, if you look at this green spot here, that corresponds to this one right over here. And so it's this whole region here that is low density in prey resources. Okay? So now I'm going to take you to 12 days later. Okay? So the 28th of May, and sure enough, the abundance of these prey has started to increase. But look at the variability. You have some locations, this one here, this green one in the middle is the middle green one here, has a density of only about 10 to 15 of these little zooplankton prey per liter or quart of water. And so that's a very poor condition for these larval fish to be in, where if they could just move over a ways, they could be in a spot where there's 5 or 6 times that amount of abundance of prey. So what we were looking for was is there heterogeneity or variability in the timing of when they come down, where these larvae go when they're in this region of Sandusky Bay, and are the prey resources distributed uniformly across the system, or are they variable? And I think these data show what we didn't expect. We kind of expected that there would be a fairly uniform distribution, because everybody looks at this system and sees it as a system that is shallow and the wind blows across it and everything gets mixed. But that's not the case. Okay? So we then go on to 2019. Chris decided he was going to stay in the upper bay here and do some sampling, but he was also going to move down to the lower bay to do some sampling. And if you look at, this is again the first day when these larval fish are present. Okay, here I'll hit this again, same thing, but the density is low again. So I think we're identifying a period of time when we think this is a very productive system, but it's that first time when those larval fish come down that in fact it could be a very stressful period for them. Okay, so that's sort of the summary of that. What we're finding, I think, is something that's really it's on a fairly small spatial scale, but it's really important because these larvae come down the system. We're sort of testing the idea here of what we call the match-mismatch hypothesis, which means do the larval fish come down the river at a time when the zooplankton, their prey, is at very high density so that they have the food available for them? That would be the match. Or is it a mismatch? They come down the river and there's not enough food around. Therefore, they don't have the resources to hang out and go on a diet for a while. They have to eat. And so they're suffering because the food's not there. Okay? This is all important because the fact that we're trying to understand the dynamics of what happens with these fish at an early life stage. And I'll point out that some of you may know, but there's a dam up in Fremont, so way up here where it says Hatch, that was taken out with very good intent, and that is to increase habitat for walleye to come up this river, find more habitat, and be able to spawn. And I think it is an excellent idea, but I have a caveat, and that is if in fact it's no longer 25 kilometers, but now 35 kilometers or 20-something miles to get down the river to the area where the zooplankton might be more abundant, are you going to get larvae that come down halfway, have used up all their food, and now need to find prey to be able to consume? They've used up their yolk, they need to find food. So that's a good conundrum that we need to explore some more. Okay? What I'm going to do now is move to Nate Stott's work. Okay? Right after I show you the hydrologic modeling that we're trying to do, because I think this is really interesting. What we are doing is we're looking at this movement of these larvae downstream in a model. It's called an EFDC model. It doesn't really matter what it is. It's a model that creates a lot of cells. It's hydrodynamic in that it monitors the currents or the movement of larvae, and larvae are really just neutrally buoyant particles. So what we're doing is we're working with Tetra Tech through Scudder Mackey and the Office of Coastal Management to try to track larval fish as they come down the river and then start to disperse. And so what we've done is just today I think Chris sent them the information, we have information of the frequency with which larvae hit this point, the mouth of the Sandusky River, and we're going to be releasing these particles and watch the movement of these larvae as they come out of the river into the bay and see if it matches Chris's distribution of those larvae. So I'm going to show you a little picture here of that. It's just the beginning of this, so it's not really good at following the exact distribution that you might expect, but you'll see some blue lines that shows the trajectory of those larval fish. There we go. So now they're starting to move out. They in this exercise, they put the larvae in different locations, or the particles. But you can see that we could begin to follow where these larval fish go if, in fact, they are neutrally buoyant and they just follow the currents that occur in this system. So I'm really excited about this. It so happens that 2015 was a very low flow year when these larvae were there, and it differs considerably from 2016 to 2018. So we're hoping to be able to see quite distinct differences in distribution patterns of these larval fish. Okay, so now I'd like to quickly move on to Nate Stott's work to show you what we've found in terms of northern pike. Nate is from Minnesota, so when he came down to work on his master's degree at Bowling Green, it was an easy move for him to start to want to work on northern pike, because that was one of the species up in Minnesota that's very, very common. Down here we're sort of somewhat near the southern limit of the distribution of northern pike, but we know that they spawn in a series of wetlands across here. All these different spots here and others are wetland areas where we can find adult pike. And so Nate decided he was going to try to look at a study to see what limits northern pike spawning success in these wetlands. Now we know that northern pike, there are two of them here, they're mating, that they follow sort of the hydrodynamics of these systems by following temperatures, but they are considered a sentinel species for wetland restoration, meaning that if you get northern pike reproducing and having offspring in these systems, it's likely that the characteristics of that wetland are good enough for many other species in there. So here are these two northern pike spawning in very, very shallow water. So in spring, and I'll show you more about that. So here's the life cycle of the northern pike. These adults come into these grassy areas very near shore. They lay their eggs, the male fertilizes those eggs, they cling to vegetation where the little embryo develops in the very near shore areas. It hatches out, it still attaches to that vegetation until it's used up its yolk, and then at that time it starts to feed, and that's the critical period again when it needs to be able to find enough food to be able to survive, to be able to grow and become one of the adults. So here's a picture of some eggs attached to vegetation. What we know is that these eggs are tolerant of very low oxygen, and you might think that because they're going to be deposited in these grassy areas which can become low oxygen, especially at night when there's decomposition occurring in there. They have to be able to tolerate very low temperatures because of the fact that the parents lay these eggs in water just after ice disappears from these wetlands. And we also have found, and this is sort of the crux of Nate's dissertation, is that we have what are called seiches. When the wind blows across the lake, we have a lake that runs mostly west to east. When we get a good blow from the west, if it's strong enough, it can push water from the western end of Lake Erie toward the eastern end. And this picture here to the right is an example of that. Nate went out one day, he sent me this picture because the day before the water in this wetland had been up into this grasses up here. These are Phragmites here. And then because the wind blew hard enough, all that water went out, so that if a northern pike had laid its eggs in along the shore here, those were now high and dry. And so that's a crux of part of his dissertation right there, but there's a lot that goes into showing that, okay? So Nate was looking for these eggs and larvae. He went and he collected a bunch of adults, over 100 of them. He was able to age them, and so what you see in the black lines here are the number of individuals that they're adults, but were alive or spawned in that particular year. So a whole bunch of individuals that he collected survived in 2012 to be caught in 2018 or 19, okay? Well, you can build a model that shows your expected mortality rate, okay? And what we see here is whenever the black column is higher than the gray column, that means more individuals survived than you would have predicted. So in 2009, 2010, 2012, we got better survival than we would have predicted. And so now we ask, well, what are the conditions that made it so good for them to be able to survive? And so we know from the literature that water level fluctuations are important, that habitat is important, that nursery habitat is after you've hatched out and you're now feeding on your own, having an environment to be able to survive in. Yes, these are all important, but how important each one is may be dependent upon the regions of the world you're in, what the environmental conditions are in. You know, are you in a wetland in the Baltic on the shore of Sweden? Are you in Russia? Are you up in Alaska? Or are you in the Great Lakes and in the southern Great Lakes where we are here? So here's an example of a sesh. You have Lake Erie here. The strong wind blows across it, pushes water toward Buffalo. And what we see is when we look at a hydrograph on the day when that occurred, and this was last year on April 13th, a good blow occurred and it drove down the water level such that it was at least a half a meter below what the average would have been. So if the mom and dad came in and laid their eggs in the vegetation and then the next day or sometime while those embryos were developing, this wind condition occurred, those eggs would have all been on vegetation still, but that vegetation now would be on land. Okay, so that seems to be the situation that's occurring here. So when tomorrow, I think it's tomorrow, maybe it's tonight, when the wind blows, it's supposed to get up to 25 to 30 miles an hour, and it's coming from the southeast. This is what will be happening out in Lake Erie, is that wind will be pushed across the western part, across Lake Erie, okay? And Nate, who has been out these past few days, knows that there are eggs now on vegetation in the wetlands of Lake Erie, so those are going to be in trouble to be able to survive when this decline in water level occurs. How does Nate know this is happening? Well, he did some work for his master's thesis where he used some high-frequency sonar. He went out, he put that down just at the time when the ice was coming off of these wetlands, sort of like this picture here where there's now some water on this wetland here. He put some high-frequency sonar in there and he tracked those adult northern pike coming into spawn. And you can see here, here's ice out down here. Let me get to my laser pointer. I like it better. Okay, and within about 3 days, almost half of all the northern pike that he saw had come into the wetland already. So they're racing in to be able to spawn just along the shorelines, just after ice is out, okay? And then a few more dribble in. Okay, so it's very early on when these guys are spawning in this habitat. However, in 2017 and 18, when he was doing this, what he found was that there was a strong spring sesh, and it looked like this. He was out trying to sample here, and he went out that morning, and this is what it looked like. And there would not be any larvae out there because there's no water. Okay, this didn't happen in 2019, okay? So in 2017, we had these 3 seshes occurring in rapid succession from late February to early March, okay? In 2018, we had another one occur in late February, okay? And so these are sort of the telltale signs that we see that they occurred likely when spawning had occurred and the water level had dropped. This did not happen in 2019. So he and some other students went out, they went sampling in 2017 and 18 and 19, looking for the little juvenile northern pike. This is now not in February, but now in June. And we're looking for these little ones. And they went into a variety of wetlands, sampled for them with seines, and what they found was each year in 2017 they found one. That's one little northern pike survived that caught it. When they went out in 2019, they caught 22 out there in actually fewer wetlands. So we know that 2019 was a reasonably good year for this species. Okay? So going back to this picture again, these 106 fish that Nate captured, trying to understand what the environmental conditions were in those years when they did well and in years when they did poorly. So what Nate did was he built an index to show how many times in a spring did you get that sesh or that water drop of over a meter? So that's 18 inches, 19 inches. And in the years when there was 1 or 2 of them, you got no reproduction at all compared to what you expected. However, in years when you got none, there were some years when they did very well, and you got large number of individuals. So this is again pointing to this idea that this oscillation in water level that you can't predict. Unlike Lake Erie, which was at its highest point last year, and now it's about a foot lower, okay, which occurs on somewhere in the neighborhood of a decadal scale, okay, this oscillation, okay. Here, you don't know if a sesh is going to come this coming year. You lay your eggs and you cross your fingers and you leave the wetland. And so we see this high degree of variability that occurs in these systems. So in 2020, Nate went out one day, I believe, and looked for those eggs at different depths. And then COVID raised its lovely head and we had to stop. So we just got some estimates of the number of eggs that we could find at different depths in the water to try to relate to these sesh activities that occur. So what he found was that they typically spawn in water less than a ½ meter deep, okay? So in these habitats, typically in these grassy areas, okay? So again, pointing to the idea that if water level goes down, like in 2017, which went down more than a ½ meter, we would expect all these eggs to be now exposed to dewatering and possibly not survive, okay? So you have a pre-sesh condition here where we put out some temperature sensors to look at temperature, and a few days later a good blow occurred and there's the same sensor high and dry up here. So just one more way of seeing that, in fact, those eggs that were in this fabulous nursery and incubation habitat would not have done well in that system. Okay, I want to get to quick measures of, because I'm out of time, wetland management, recognizing that our goal is for nutrient retention. When we're trying to manage these wetlands, we also often do it to help to maintain waterfowl, migratory birds, other aquatic organisms from amphibians to reptiles, and now we're adding northern pike to this mix. So wetland managers have a lot of juggling to do, okay? If there's a natural system, you get natural oscillations that occur, but if you're having a managed system here, this is one in California, you have the opportunity to adjust and maintain different water levels in your system, and I'm just suggesting that we try to figure out how to add northern pike's needs in early spring to that mix, because if you could keep the water from leaving when a wind blows hard from the west, you might be able to preserve those eggs that have now been laid in shallow water and be able to get better survivorship of those young. Okay, so we're at the point where I've pretty much done. Here are some acknowledgements, lots of different groups from the Department of Natural Resources in different areas to different societies that have helped to support Nate, to Ottawa National Wildlife Refuge, the Nature Conservancy, a whole bunch of students who have helped out, Kevin Kratt and Ben and Sen Bao, and also our own Center for Great Lakes and Watershed Studies. So I'm done here. I'm happy to answer any questions and bring in the other people. I'm going to leave a set of questions up there for you to ask each of the speakers, which I know they would love to address. So.
Wonderful, thank you, Jeff. So I'm Kevin McClooney. I'm an associate professor in the Department of Biological Sciences here at BGSU, and I will be the moderator for the question and answer session. Just a few things to go over. So raise your hand if you have a question. What I mean is your virtual hand. So if you look down at the bottom of your screen, you should have a button that says reactions. Might look a little different if you're on a phone. And then there should be a button that says raise hand. So if you do that, then I'll look and I'll see that and I'll call on you when it's your turn. Alternatively, you can put your question directly in the Zoom chat window. So down there next to the reactions button, or possibly where it says more, there should be something that says chat. And if you open that, you can type your chat message in. You can either send it to everyone, or you can even send it just to me. And I can read the question, and then the panelists can answer. So now I'm going to turn it over to Jeff to introduce his panelists.
Okay. Today we have Dr. Scudder Mackey, who is the chief for the Office of Coastal Management for the Department of Natural Resources. Is Scudder on screen? Yeah, great.
Yep.
I see him, but I don't know if everybody else does. And Scudder is a hydrologist. Am I right, Scudder? Or do you call yourself a geologist? Maybe a geologist.
I'm a geologist, but
okay.
I do a lot of hydrology.
Yep, exactly. And so he has been instrumental in pushing forward with restoration activities, primarily in many areas across the Lake Erie. But since we're talking about Sandusky Bay today, I'd say the Sandusky Bay initiative to try to improve the water quality in Sandusky Bay from a variety of different ways. And so I'm really happy to have Scudder here tonight for you to ask questions. And then there's Chris Kemp, who's a PhD student of mine. Chris, you want to raise your hand? I don't see him raising his hand. Okay.
Right here.
There he is. Okay. Chris did that larval walleye work, which I think was really, really top-notch stuff. And Nate Stott is also a PhD student working with me on the Northern Pike, finding some things like that sesh activity that I think people we in some ways have had a hard time convincing people of the importance of that. So I'm really proud of that work also.
Wonderful, thank you. So people that have been coming to these for a while know that I often like to start off with a similar question to the following one. Get us going. How did you become a fish biologist? What got you interested? And this could be for all the panelists, really, I think. But we'll start with Jeff.
Okay. There was a creek at the bottom of my street. And I suspect that what I remember is my mom calling us for lunch and for dinner, because we'd be down there all the time. And so I'm not so sure it's fish, but it's aquatic biology, which has always attracted me. So that's it.
That's the short version. I know that you I could tell a much longer version of your path here. Huh? How about Nate? Do you want to go next?
Yeah, sure, I can go. So for me, it seemed like a pretty natural progression. So I'm just a kid originally from Minnesota, and actually my grandparents owned a fishing resort on Mille Lacs Lake. I don't know if anybody's heard of that, but it's a 240,000 acre lake. And so I always grew up in and around that resort. And so it just seemed like a very natural progression for me to be moving into the exact opposite side of the field, all relating to the fish. So that's really where that came from.
And I'm going to tell you a story about Nate, because Nate was about 7 or 8 years old, and there were no other guides to take fishermen out. And some fishermen came up and they wanted a guide. And I don't know if it's your grandfather, Nate, but you turned to Nate and you said, oh, Nate here can do it for you. And they kind of looked skeptically at him, but they took him on, and they came back with a boatload of fish that day. So I think he, from about the age of 7 or 8, he's been a fisherman at heart.
Yeah, that did happen. Yep, I like that story, but it's a good one.
Are you still bringing home boatloads of fish?
When I get out fishing, yes. It's less often nowadays. You read about fish all day, you don't go fishing nearly as much.
I get it. Chris, how about you?
I sort of came to the game later than I think probably these guys. I was always interested in biology, kind of started out in herpetology and entomology, working for a natural history museum, which was a lot of fun. But then I got a job for the USGS out in the hills in like northeast Oklahoma. And I was pretty much hooked. We were working on summer movements of a bunch of different species, and I got hooked looking at all the different fish species that were living in these crazy streams, and decided to end up in graduate school. That kind of got me hooked.
I love asking this question because it's often different. Not everybody got here the same way, right? Scudder, I don't think you really consider yourself a fish biologist per se, but can you tell us a little bit about how you got interested in science and management? And
Yeah, sure. Yeah, thank God I'm not a fish biologist, so I'll just leave it at that. And actually, what got me started was an 8th grade earth science course. Really took to it. And I'm more of a physical scientist. I'm interested in a lot of the physical processes. And so that's the reason I went into geology. I would say that, you know, tying it back to the fishery, though, what I'm really interested in is how physical processes actually create habitat structure and influence the biological systems. And so the type of work that Nate and Chris have been doing are very much tied to the interactions of hydrology and substrate and, you know, the physical processes which are actually driving the biological systems. And that's where I think with geologists we can partner and work very closely with the biologists to get a better understanding of how these systems actually function. So.
That's a good point. And I think that illustrates how a lot of us biologists need to collaborate with other kinds of scientists to do our job well, at least environmental biologists for sure. Does anybody from the audience have a question? I see Tim has his hand raised. Tim, do you want to go ahead?
Sure, I thought I'd just start off. So Jeff, great presentation. Thanks a lot. You said something at the beginning of your presentation that I just wanted to follow up on, because you talked about the two populations of walleye, right? The reef spawners and the river spawners. You're focusing mainly on the river, but could you just go into that a little bit more in terms of, do you see any crossbreeding? Do reef spawners always go to reefs, or is there a mixing of reef and river, or is it what's closest or most convenient? Do we know?
It's more than just what's close and convenient. There's been some people down at OSU. We've done some work with white bass and found that there is reasonable fidelity. Reasonable meaning, let's say that 70% of the individuals who were spawned in a location come back to that location. And so you do get some mixing. It's not like, you know, steelhead salmon in the Pacific Northwest, where a very high percentage, 98% of the individuals come back to their same place where they were spawned. But it's pretty good return. And so one of the things that Nate was doing was, we have a technique. We take a bone in the fish and we can run a laser across it, and there are certain chemicals in there. And using the Sandusky system as an example, there is an unusually high amounts of strontium in the water because of the bedrock around the Sandusky River watershed. And so if a fish is raised in that area, the amount of strontium to the amount of calcium in their bones is very high. And so we can tell with almost complete certainty that a fish, when it's an adult, whether it was born in the Sandusky River system or someplace else. And the Maumee is a little lower, and the reefs get most of their water from the Huron River, so they are much lower in that strontium. So you can begin to sort of tease out who's who and where they've come from. Hopefully that answered your question a little bit.
Yeah, absolutely. And then I just had a quick follow-up, just with, you know, we're hearing a lot from the sport fishing and the charter boat captains that there's just a lot of walleye on the lake right now. Do you know what's kind of responsible for that? You know, such the high, just sheer numbers of walleye that are out in the lake?
Yeah, I think I can't remember the exact year classes, but it's either the 2017 and 18 or 18 and 19 year classes were, well, I know the 19 was just phenomenal, possibly close to the highest they've ever seen. And so those fish from 2019, say in 2021, are about 14, 15 inches in length. And so they've now what we call recruited to the fishery. And so they're coming in to get ready to spawn themselves, or at least come to the dance, get ready to spawn. And so there are all these millions and millions. I can't remember if it's 60 million or more adult walleye that they're having now. So now they've been able to increase the catch. Now in the rivers you can keep 6 fish per day. You can keep 6 fish if you're out on the reefs, because they know that they've got so many fish, and it's just chance. At this point in time, you have to say it's chance that you had a good year, and a couple of good years. There are reasons for it, but whether they can be repeated, it seems to occur periodically when you get a good year class.
So let's see, I've got another question here. If larval fish come down the river at the wrong time for getting food, is there any management that could be done to help them?
No. Not really in this case. They're just floating down the river, and I don't see, it's not like you're going to feed them or you're going to try to increase production of the zooplankton that's available to them. That's one of the dynamics that occurs here. So let's say they start to come down the river and you have a good amount of rain, and now instead of taking 4 days to get down the river, it takes 2. Well, now maybe they've been blown out enough into Sandusky Bay that they happen to encounter and get mixed in with those higher productivity areas. And so maybe they'll do fine. That's why I think the 2015 hydrodynamic information is really going to be interesting, because there's so little water coming down the river that these larvae may get sort of stuck in that very western location of Sandusky Bay and not be able to get into that mixed higher productivity area.
Great. Unless any of the panelists have anything to say about that, can do another question.
Well, to follow up on that, the fisheries, Ohio Division of Wildlife Fisheries, Sandusky office, just a couple of blocks, at least from our old office. We've relocated in Sandusky now. And obviously they're very pleased with the, with the really good walleye fishing. It's really good for the state. It's good for recreation. And I think that they think that there is a pretty good linkage with the high water events that we've had. And I think, Jeff, when you have high water events like this, high lake levels, that usually means increased precipitation in the basin. And that means, more frequent storms. And now we're starting to get more into the climate side of things. But if you have more frequent storms, as long as the discharges aren't excessive, you may be moving the larval fish down through the system a bit more quickly. And as you say, if Chris's distribution of the prey is correct, then maybe you are shooting these guys out further into the bay, and they're actually more successful. And that's one of the reasons why you might see a larger year class. But again, I don't think you can prove it at this point, but I think that's probably some positive speculation. And I know the Division of Wildlife is thinking along those lines as well.
I'll just introduce the idea. Mark DeFore, who got his PhD at University of Toledo, did a wonderful research project where he was looking at the relative contribution of these walleye from these different sources, the river systems, the reefs out in the lake, and pointed out very nicely that in fact what we have is what most people would say when you're holding on some stocks. You want to diversify as much as possible. And so this has been coined as the portfolio effect, in that maybe you have years when in fact the numbers of larvae that get produced in the Sandusky and the Maumee is low for whatever environmental reason, but the reef condition is just great. Well, that gives you a good contribution from the reef population, but maybe next year it's the river systems that do well and the reef doesn't do so well because of certain storm events, or as Scudder said, lake level. And so it's this idea of having diversity of different locations where they can reproduce allows for some group to do well. And that's going to lead to a more stable population in the long run.
So I think that you may have just answered the next question, which was, I will ask it anyway and see if you have anything to add. Could climate change be influencing when the larvae move downstream? Could climate change make the mismatch worse or better? So that's kind of, I think you basically already answered that, but do you have anything to add?
Yeah, Chris has something to add to that. I'm going to shut up.
I think we've already hit on some of the things that might be affected by climate change. It is interesting that if you look at some of the work from the early times, the 90s, and even the last 2 years that we've been doing this, the hatches do seem to be occurring slightly later in the year, and we've seen that in. I think Nate has seen some things that are occurring later in the year as well. So I don't know if discharge events are greater in magnitude, could fire them out closer to the Edison Bridge, which might be, I would say, more of a match, right? Rather than holding them in the far western side, which is where we are thinking is the mismatch. I'm not sure. The short answer is it depends. It's hard to tell what the direct effect of climate change would be on larval fish here.
And Nate,
Okay.
and I'll just let Nate speak up, because he's kind of looking at this idea of climate change for his northern pike.
Yeah, so for when it comes to my my northern pike, they're very, very tied to when the ice is actually coming off of some of these wetland systems. And from a lot of the work that I've seen, at least within Lake Erie, which, the last time that a whole lot of people were looking at northern pike, they were really looking at somewhere in early April is when these northern pike were spawning, and because that was coinciding with the early April ice out. Whereas in the 5 years that I've been here, I have seen an ice out as early as February 11, and then as late as a March 22, with the spawning being basically March 26 or being as early as February 15, 16. So I'm seeing very variable times when it comes to spawning. I mean, just this year, we had something like 8 inches of ice on the 1st of March in some of my sample sites. However, within about a week and a half, what I'm presuming almost all of the northern pike had already spawned. So we just had a very, very quick spring this year as far as, my northern pike spawning. Whereas, last year we had an ice out at the beginning of March, pretty close to the same time period as now. But that spawn was actually almost a 3.5 week window before they had completely spawned out, whereas this year was only a 3-day window. So I'm just seeing lots and lots of variability is about all that. That's the biggest proponent that I could really, figure. Expect the unexpected, if you will.
Thanks. I think we'll just have one more question before we formally conclude. Given that seiches have a fairly characteristic pattern on water level graphs, are these random, or is there a pattern emerging over history?
I suppose I'll take that one. At least for the highest magnitude, it is definitely not random, but it is 100% related to weather. I don't know if anybody's been looking at the weather for what tonight's supposed to be, but we're supposed to get gusts up to 60 miles an hour from the southwest. So I'm expecting, if that is actually happening, we might have somewhere between a 0.8 and a 0.9 meter sesh tonight. But as of now, the wind hasn't picked up, and it was supposed to have started an hour and a half ago. So we'll see about that. But also, when it comes to the sesh, you have other things that are all classified in that. And Lake Erie, as far as I know, I think it's 4. Scudder, you could correct me if I'm wrong, but there's 4 different periodicities when it comes to a sesh, and it determines based on the sloshing after that first event. And they have very different times. It's like a 6, 8, 12, and 14 hours. as far as different times of the water will basically slosh back and forth after that initial, storm or wind event occurs. And those will last for weeks on end, where you just have this very, almost like, I don't really know what the word is, but it's almost like a stable oscillations based on those different periodicities, where it'll keep going up, then dropping down, and each time just get a little bit less in magnitude. But how we're really looking at is we're really looking at those, that first event, which is, it's weather dependent, not necessarily random.
Following up on Nate's comment, he's right. And a couple of things to know: a real strong southwest wind. Some of the larger sesh events, you can have a 16-foot or roughly a 4-meter difference in water surface elevation between Toledo and Buffalo. So that's a tilted lake surface. And when that wind stops, it's like tilting a bathtub and then watching the water basically slosh around the back, just around the bathtub. And that's the periodicity that Nate's talking about. We have what they call an ADCP current profiler underneath the Edison Bridge. It's part of some other work that we're doing in Sandusky Bay. And we see basically it's about a 14.2 hour period where we actually get flow reversals underneath the Edison Bridge. Water flows into the inner bay and then out of the inner bay into the outer bay at about every 14 hours. And so it gives you an idea of some of the periodicity and some of the variability that we have in water levels. Again, those water level fluctuations are only a handful of centimeters within the bay. We're talking about the major wind events that really could drop water levels, as Nate indicated, half a meter or more. And those are the ones that are probably most impactful in the wetlands that he's working. So, yep.
Great, thank you. I'm going to let Tim take over for doing sort of some final words.
Well, thank you. I just wanted to thank our, especially Dr. Miner and our panelists again for a great, talk and a great discussion. Thanks everyone for